All attendees are in listen-only mode.
Hello everyone and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar that is organized by the GastroPlus user group. Today's presentation is titled Use of Oral Absorption Modeling to Characterize Drug Release and Absorption of a BCS II compound from immediate release formulations. This presentation will be given by Dr. Cordula Stillhart from Hoffmann-La Roche Ltd. Dr. Cordula Stillhart is working as a biopharmaceutical scientist at Hoffmann-La Roche Ltd, Basel, Switzerland. She holds a Master in Pharmaceutical Sciences and obtained her Ph.D. from the University of Basel, where her research focused on the biopharmaceutical behavior of oral lipid-based formulations. After her Ph.D. studies, she joined the Roche Formulation Research and Development Group as lab head and a biopharmaceutics expert for small molecule development projects. Also participating as a panelist today is Dr. Vera Lukchova from Formulations Plus. Vera is the team leader for simulation technologies and also serves as the GastroPlus product manager. She will be available to answer questions related to the GastroPlus program. This webinar will be recorded and it will be available to the user group members in the future. Following the presentation, there will be an opportunity to ask questions. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. This enables us to unmute your line so that we can hear you. So without further ado, we now begin the webinar presentation. Cordula?
Yeah, many thanks, Sherlyn, for this introduction, and welcome to today's GastroPlus user group webinar from my side as well. During this webinar, I will present a recent application of oral absorption modeling during formulation development of a BCS II compound here, which we conducted here at Hoffmann-La Roche. We used GastroPlus to gain a deeper understanding about the mechanism of oral absorption and to understand the critical API and formulation parameters which affect oral exposure. This is a brief outline of my presentation. I will start with some background information about the CMC properties of the API and the formulations we used in clinical studies. And then I will summarize the clinical pharmacokinetic behavior of this compound, which we observed in clinical studies so far. In the second part, I will describe how we characterize the drug release and absorption behavior of our compound using oral absorption modeling in GastroPlus, and how we further try to understand the biopharmaceutical behavior using in vitro dissolution testing and Raman imaging studies. In the last part of my presentation, I will then present the development of an IVIVC, an in vitro in vivo correlation model for future formulation development of the present compound. So starting with some background information, our development compound is called basmisanil. It is an oral available low molecular weight compound. It has a basic pKa of around 2, and it exhibits moderate lipophilicity with a log D of around 1.9. Solubility is generally very low. The measured values in aqueous buffer solutions were all below 1 microgram per ml over the entire physiological pH range. In the presence of biosalt micelles, solubility was slightly higher, and we obtained values of 10 microgram per ml in FASIF and 32 microgram per ml in FASIF. Permeability was shown to be high, and therefore, based on solubility and the good permeability values obtained in vitro, the compound was classified as class 2 according to the Biopharmaceutics classification system. The API was formulated in its crystalline state, which was a stable crystal, and in solution as well, we observed good stability over the physiological pH range. The formulations we used in clinical studies so far were standard immediate release oral formulations, and the formulations were actually quite similar, so there were minor changes during clinical development. In phase 1 clinical studies, we used an immediate release tablet formulation, which was provided in as 4 different dose strengths, and the tablet was a same size tablet with increasing drug load from 0.5 up to 250 milligram. In phase 2 clinical studies, we used a film-coated tablet formulation, which was an immediate release formulation as well. The tablet composition was very similar to the phase 1 tablet formulation, and the film coat was a non-functional film coat which dissolved very rapidly in aqueous solutions. Apart from the tablet formulation, we also introduced a granules in sachet formulation, which was also an immediate release drug product, and both formulations used in phase 2 clinical studies exhibited a dose strength of 120 milligram. Here we see an overview of the formulation manufacturing process of the tablets, the film-coated tablet, and the granules in sachet formulation, which we used in clinical studies. The API was formulated in a micronized form. The D90 of clinical batches was around 10 micrometers. So the API was mixed with standard excipients, including filler, binder, disintegrant, and lubricant. In the phase 1 tablet formulation, we additionally included a surfactant. However, we observed that it provided no additional benefit granules in sachet formulation. For preparation of the tablet formulation, the granules were further mixed with the excipients, standard excipients representing the exogranular phase, and then the final blend was compressed to a tablet. As I mentioned before, phase 2 tablet formulation was additionally provided with a film coat, which was shown to have no influence on the drug, on the disintegration or the dissolution rate of the final drug product. Here we see an overview of the clinical pharmacokinetic profile we obtained in phase 1 and phase 2 studies for the tablet formulation, as well as for the phase 2 film-coated tablet and the granules in sachet formulation. So on the left-hand side, we see the results from signal ascending dose studies. which showed that the exposure was dose proportional up to a dose of around 130 milligram. And for increasing doses, the exposure was shown to be less than dose proportional for Cmax and AUC. During phase 2 studies, we obtained comparable exposure from the granules in sachet formulation as we obtained earlier from the tablet formulation, but the film-coated tablet, which exhibited the same dose strength as the granules in sachet formulation, resulted in a comparatively lower exposure compared to the granules and the earlier tablet formulation. So the objective of our studies was, first of all, to characterize the mechanism of drug release and absorption from these different immediate release formulations, to better understand the rate-limiting steps of absorption. A second objective was to understand the root cause for the different exposure we observed following administration of the film-coated tablets used in phase 2, to learn about the critical API, formulation and process parameters which affect oral absorption of basmisanil. And finally, the third objective was to develop an in vitro in vivo correlation model which we could then use for future formulation development. So as I mentioned before, we started with the characterization of drug release and absorption using the clinical data we got so far by developing an oral absorption model in GastroPlus. So this is just a brief summary of the input parameters we used in the oral absorption model. So compound properties corresponded to the experimental values, physicochemical properties we determined in vitro, as for example, permeability values, solubility in aqueous media and biorelevant media, and so on. The kinetics of drug release was simulated by assuming an immediate release tablet formulation in case of the tablets and an immediate release suspension in case of the granules formulation. The dissolution model we applied was the default Johnson dissolution model. The single ascending dose studies, as well as the pharmacokinetic studies done in phase 2, were all conducted under fed state conditions. So for this reason, we use the human physiological fat gut physiology and the default absorption scale factor model included in GastroPlus. The disposition PK was described by fitting a 2-compartment PK model to the intravenous microdosing data we obtained in an earlier absolute bioavailability study, and this has been done using the built-in module PK Plus in GastroPlus. So first step was to verify our model using the clinical data obtained from the tablet formulation in phase 1 studies, which were the single ascending dose studies. So here we see the observed and in blue the predicted profile, plasma concentration time profile for, as an example, a single 160 milligram dose administration, which has been administered as a tablet formulation. And we can see that the model is very well predictive for the observed plasma concentration time profile. We verified the model accuracy over the entire dosing range, and the result is here summarized as dose normalized Cmax for the entire dosing range between 1.5 and 1250 milligrams. And we can see that again in blue, the blue profile shows the predicted data, and the marks here are the observed values. Again, the GastroPlus model resulted to be very predictive for the observed data, and especially it was shown to describe very well this dose proportionality in Cmax and AUC as well, which we observed up to a dose of around 120 milligram, and the decreasing dose normalized Cmax for higher doses. So given this good agreement between the observed and predicted profiles, it was reasonable to explore the mechanism of this dose proportionality in drug absorption using the oral absorption model. And this is the next thing we did. We analyzed the intraluminal drug concentrations for a dose range which was in the dose proportional range with regard to Cmax, as well as one dose which was here at the borderline between where we see this decrease in dose normalized Cmax, as well one dose which was which exhibited significantly lower dose normalized Cmax compared to lower doses. Here we see the drug concentration profiles as simulated with our absorption model for the 3 different doses. So for the lowest dose, we see that the intraluminal concentration profiles are very low, and if we compare this value with, for example, FASIF and FASIF data, which were higher than 10 microgram per ml, we can conclude that for this lowest dose, solubility doesn't represent a limitation for absorption. However, when we go to the next higher dose, we are in a much higher concentration range for the 120 milligram. And when increasing the dose to the 330 milligram, we see that there is not much of an increase in intraluminal drug concentration. So this suggests that beyond this dose, we are most likely in a dosing range where solubility becomes the rate limiting step for absorption, while below the dissolution rate the oral absorption model for the 2 formulations we used in phase 2 studies, which were the granules in sachet formulation and the film-coated tablet formulation. The granules in sachet formulation again resulted in a very good prediction of the observed plasma concentration time profiles. But as expected, for the 120 milligram film-coated tablet formulation, we observed a significant overprediction in Cmax and AUC using our oral absorption model. So we wanted to understand which formulation properties resulted in this comparably lower exposure. And to this end, we conducted a parameter sensitivity analysis, which showed that the drug particle size was actually very sensitive parameter. So again, here is the result for the three different dose strengths we selected for this analysis. The lowest dose doesn't seem to be sensitive in the drug particle size range we use in clinical formulations. But with the 120 milligram formulation, we see that the API particle size we used in clinical formulation is in a very sensitive range with regard to its impact on the absorption rate. For the highest dose we tested, we know that in this concentration range, absorption is very much limited by the solubility of the API in the intestinal fluids, and therefore the impact of particle size is less significant. So to better understand which formulation and or processing step was the reason for this decrease in dissolution rate, we analyzed the in vitro dissolution profiles of phase 2 formulations as well as of the drug product intermediates in more detail. To this end, we conducted in vitro dissolution studies in passive media at a constant concentration of corresponding to 40 milligram of API in 900 milliliter of passive. And this concentration allowed almost 80% of the drug compound to be solubilized before reaching the solubility limit, and therefore we were able to discriminate with regard to the dissolution rate. As we can see from this plot, the granules in sachet formulation dissolved much faster compared to the 120 milligram film-coated tablet, which is the lowest curve here. Further, if we compare the 120 milligram granules in sachet formulation with the corresponding intermediate drug product intermediate for the 120 milligram film-coated tablet, we already see that the formulation composition has some impact on the dissolution rate, since the corresponding granules dissolve already much slower compared to the granules in sachet formulation. A further decrease was then observed when processing the granules into the final blend and especially into the final film-coated tablet. So from this data, we learned that the manufacturing process as well as the formulation composition had some influence on the dissolution rate of our compound. As I mentioned earlier, the formulation we used in clinical studies were very similar with regard to the manufacturing process and also with regard to the qualitative composition of the formulation. However, if we compare the quantitative composition of the formulation, we see that there is a difference with regard to the drug load. So in the phase 1 tablet formulation, we have comparatively low drug load for the dose strength between 0.5 and 40 milligram, but quite high drug load in 250 milligram tablet formulation. And as well, when we compare the granules in sachet formulation with the film-coated tablet formulation, we see that the drug load is comparatively high in the film-coated tablet formulation. And we know from the theory that beyond a certain drug load, there is a risk of exceeding the percolation threshold in the tablet matrix. So what does this mean exactly? In an ideal formulation, we should have the API, which is shown here schematically in blue. The API particles should be distributed as individual clusters in the tablet matrix, which is shown here in yellow. However, if we increase the concentration of API in the formulation beyond a certain critical concentration, we may reach the so-called percolation threshold, and at this point the API particles start forming a continuous network of API within the tablet matrix. Now, if a tablet formulation disintegrates as long as we are below the percolation threshold, we can expect the API to be released as single individual API particles. However, if we have tablet disintegration beyond the percolation threshold, the API particles may not be released as single micronized particles anymore, but rather as larger aggregate of multiple API particles. As a consequence of that, of course, the API surface decreases, and in turn the dissolution rate as well. And we know from the parameter sensitivity analysis, which we have done with our oral absorption model for basmisanil specifically, that if we, for some reasons, we change the drug particle size and therefore this API surface area available for dissolution, we have directly a significant impact on the Cmax of our compound. To further verify our hypothesis that we may have exceeded the percolation threshold in our film-coated tablet formulation, we conducted Raman imaging studies of granules, the granules formulation, tablet and film-coated tablet formulation. So what we see here are Raman images which show in red the drug substance and in black the formulation matrix. Here on the left-hand side we have the image for the granules in sachet formulation. In the middle is the Raman image for the tablet formulation with a 40 milligram drug load, which was beyond, which corresponded to a low drug load. And on the right-hand side, we see the film-coated tablet can be explained by the cohesive properties of the API, which shows some tendency to form aggregates in a bulk material. However, when we compare the granules in sachet formulation with the tablet formulation, we see that the tablet compression step further increases this cohesion of API particles. Here in the 40 milligram tablet formulation, we see some regions of high drug particle density, but these regions are do still include some excipients which can be seen as black shaded areas. When we increase the drug load, however, we see that the API starts forming a large network of agglomerated clusters, and within these clusters there is almost no excipient anymore. So therefore, using based on this Raman imaging data, we could further show our hypothesis or confirm our hypothesis that after disintegration of the tablet formulation, we may have obtained larger agglomerated clusters instead of single API particles. So what we learned from this first part of oral absorption modeling studies was that we identified those ranges which exhibited dissolution and solubility limited drug absorption below and above around 130 milligram of basmisanil. We learned that formulation changes which directly affect the dissolution rate of the API have a direct impact on the oral exposure for doses which are below around 130 to 160 milligram. And we could understand that the percolation threshold was exceeded in case of the 120 milligram film-coated tablet due to the combination of several factors. On the one hand side, the high drug load in the formulation, together with the cohesive properties of the API, as well as the application of compaction and compression processes, which further induce the API to form agglomerates. As a consequence of this, the aggregation of micronized API within the tablet matrix, we observed the slower drug release and therefore the reduced absorption of the compound in vivo. The impact of the drug load on the oral exposure was less pronounced with the 250 milligram tablet formulation, even though we had a very high drug load there as well. And this is most likely due to the fact that at this high dose, in this high dose range, we are already in a range where we can observe solubility limited absorption, and therefore the impact of particle size or total surface area on the drug absorption rate was less pronounced. So now we have seen that our oral absorption model provided a very good prediction of oral exposure. And this prediction was particularly good for doses below 160 milligram, as long as the formulation released the primary API particles after disintegration. The model prediction was, however, comparatively poor when we had API aggregation during, as a consequence of tablet compression, as we have shown, for example, with the film-coated tablet formulation. And in this situation, of course, the development of an in vitro in vivo correlation provides a clear benefit because it captures the release profile of the entire formulation, and therefore it includes a potential influence of excipients or of individual process parameters on the release profile. So for this reason, we wanted to develop an in vitro in vivo correlation model, and this is now briefly described here. So we developed the IVIVC model using the IVIVC Plus module in GastroPlus. We used the in vitro dissolution data we obtained for the granules in sachet and the film-coated tablet formulation for the IVIVC model development. As I mentioned before, these data were obtained in a USP2 pedal dissolution apparatus using FASIF as a dissolution medium and using a constant API amount of 40 milligram. The corresponding in vivo data we have already seen before were used for model development, and since we used in vivo data which were obtained under fed state conditions, we used the GastroPlus mechanistic absorption method as a deconvolution method. And for comparison, we also tested the result using the traditional deconvolution method, but of course, in case of when using fed state data, it is more appropriate to use the GastroPlus mechanistic absorption method. Model verification was then done using the in vitro data obtained from the phase 1 tablet formulation, including the different dose strengths and the corresponding in vivo data over the entire dosing range from 1.5 to 1250 milligram. So here we see the percent dissolved in vitro versus the percent dissolved in vivo as obtained from the IVIVC model, and we see that there is a very nice correlation following the convolution using the GastroPlus mechanistic absorption method, and we were able to describe this using a power correlation function. This model was then applied to the entire data set we obtained from SID studies, and as is summarized here, we see that we obtained an excellent prediction over the entire dosing range. The filled symbols represent the phase 1 study data which were not included in model development, and here the empty symbols represent the data sets which were used for model development. And yeah, in this case we see that compared to the simulations we obtained before, in case of the film-coated tablet formulation, now we obtain a much better correlation with in vivo data using the IVIVC model. Yeah, so to summarize the learnings from these studies, class 2 compounds, and the elucidation of the mechanism of drug absorption and this better understanding of the critical factors which affect the drug absorption behavior, which may be related to the API or to the formulation itself, allow rational and science-based formulation development according to the quality by design strategy which we apply during formulation development. And finally, we observed that the GastroPlus mechanistic absorption deconvolution method enabled the development of a powerful IVIVC model, even in that case where we had the administration of a compound in the fat state and which exhibited a dose-dependent absorption behavior. Yeah, and finally, if someone is interested in more details about these studies, I'd like to refer to a recent publication in the AAPS journal where we actually presented the results of these studies. And before concluding my presentation, I would like to acknowledge all my colleagues here at Hoffmann-La Roche who provided input to these studies. And of course, I would like to thank you all for your kind attention. And now I'd be happy to answer your questions.
Okay, thank you very much, Cordula, for the great presentation. At this point, we will open up for questions. I'll start with the first question.
Mm-hmm.
Was the intraluminal concentration experimentally determined or estimated by the GastroPlus model? I think this is referring to your slide number 12.
Right, yeah. The intraluminal drug concentration as shown here were the simulated concentrations according to our oral absorption model.
Yeah.
So we didn't directly measure the solubility in intraluminal fluids.
I'll add to that question. If you look at the high dose 330 milligram one, the duodenum concentration, the highest point reached to over 15 microgram per ml, and the solubility in passive, and in your earlier slides shows around 30 something microgram. Yeah. So this suggests that the drug is super saturated in the duodenum for this case here.
Could be due that there is local supersaturation, but I guess this might also be due to a higher local concentration of bile salts, which then results in a higher dissolution, extent of dissolution of the compound.
So this formulation is a conventional formulation. There are no, it's not solubilized formulation. I was wondering if this is true. What is causing this supersaturation to begin with? I'm not sure you have the solubility is pH dependent. Do you have a higher solubility and a low pH? And that could create the supersaturation. If it's not that case, wondering, is it just the program that give you, because this is calculated, it's not the experimental one.
Right. Yeah. Yeah, I think temporaneous supersaturation might occur due to initial dissolution of the compound and then due to potentially faster absorption of intraluminal water, which then leads to certain supersaturation, even in the case of a conventional dosage form.
Okay, thank you. The next question. Was food effect observed in human trials, given that the drug solubility in passive is 3 times that in passive, FASSIF?
Yeah, it's a good question. Indeed, we observed a significant food effect for this compound, but we in our oral absorption modeling studies, we focused on fat state data simply because we had more clinical data available to verify and further study the oral absorption behavior of this compound. But yes, we did observe a significant food effect for this compound, as expected for the differences in fasted and fat state simulated intestinal fluid solubility data.
Yeah, I guess the same individual asked another question, is why use passive at the dissolution media instead of passive?
We used passive simply because the solubility is higher, and therefore it was, from a practical reason, more appropriate to gain nice dissolution profiles, while when using fasted state simulated intestinal fluids, we rapidly reached the solubility limit, and therefore it was more difficult to discriminate between dissolution rates.
Thank you. The next question, there are actually two questions lumped together. The first one is, there are any other methods of characterization that can be used before going for sophisticated Raman imaging? That's first the question. Second question is, can any other model be selected or next suitable other than mechanistic absorption deconvolution method? There's two questions from one individual.
Yeah, so if I'm not sure if I understood correctly the question. The first question is whether there are other analytical methods which might be used to
characterize other than Raman imaging.
Yeah, I'm not an expert in solid state characterization, but I guess depending on the API properties, there might be other techniques, such as, for example, NIR imaging or similar methods which could be used for such kind of imaging studies. But yeah, again, it depends on the API properties, which method is more appropriate for this kind of studies. And with regard to the second question, may I ask you to repeat it again?
Sure. Yeah. Can any other model be selected other than mechanistic absorption deconvolution method?
Yeah. So we, for comparison, we tried to develop an IVIVC model as well with standard deconvolution method, as for example the numerical and the low Riegelmann deconvolution method, but yeah, these methods are more appropriate for developing IVIVC models for fasted state conditions and for API which show linear absorption behavior. And in this case, where we had to develop an IVIVC model under fed state conditions, it resulted to be much more appropriate to use the mechanistic absorption deconvolution method.
Thank you. The next question I recognized from Natalie. Was the IVIVC presented to the health authorities? Has this been presented to the regulatory agents? That was the question. I'm sorry. Hello?
Charlene?
Yes. Did you hear the question? Sorry.
Yeah, no, I couldn't hear the question. I'm sorry. Could you maybe repeat it?
Yeah, sure. The question is, has the IVIVC presented to the health authorities?
No, the IVIVC has not yet been presented to the health authorities. It has been used so far during formulation development to provide guidance on potential influence of formulation changes on the oral absorption, and yeah, in the framework of ongoing formulation development, but not yet for interactions with health authorities.
Okay, thank you. Can you go to slide number 22? The question here is on slide number 22. What is the time point for the percentage dissolved in vitro?
The time point for, well, the in vitro dissolution profile has actually been modeled using a single Weibull function. So it includes all individual dissolution data and not a single time point.
Yeah, it is buried. It will not be showing in this type of plot. Okay, thank you. The next question. What is the best alternative to determine systemic clearance parameters if the IV data for drug is not available?
Well, in our case, we had, well, I have to go back to my slide. Here. So in our case, we had intravenous microdosing data available, based on which we could determine the disposition PK by fitting a two-compartment PK model. Other methods might include like more mechanistic determination of clearance, including a full PBPK model, for example. But of course, it becomes more accurate as long as you have intravenous data available.
Thank you. The next question, slide number 21. Not sure this is the, maybe you need to clarify. The in vitro data use 40 milligram, indicate the API amount 40 gram. The in vivo you have 120 milligram as a strength. The question here is why did you use the 40 milligram strength for the dissol data to establish the IVIVC? Is there not a risk to have different cluster effect at 40 milligram compared to the 120 milligram strength in vivo data used for the IVIVC? I wonder if you're just showing this as an example and not really direct correlation, or maybe you can comment on.
Yeah, right. So we use a constant amount of API in order to have always the same, well, how can I explain?
Same drug amount for the in vitro and also in vivo, or
no, so we used, for in vitro data, we used the 40 milligram API, which was indeed different from in vivo data, where we had the entire 120 milligram formulations. However, we wanted to have in vitro the same saturation limit for the different formulation in order to discriminate between different dissolution rates. And at the end, the in vitro data are expressed as a % of drug dissolved, and yeah, this can be reflected to different dose strengths at the end, even though we use a constant API amount in vitro studies. Does this answer the question?
we will, yeah, if, let's see who asked this question here. And Sophie, and if you want a further clarification, you can enter your follow-up questions. I'm going to move on to the next questions. Did you take Raman image on after this dissolution liquid sample or solid formulation sample?
No, the Raman images we collected were of the dry formulation before being tested in any in vitro dissolution test, and yeah, we did not further analyze the formulation after disintegration or during dissolution testing.
Thank you. Is there any release in stomach that may cause temporary supersaturation in duodenum? This is probably related to the earlier discussion.
Yeah. So according to the aqueous solubility data we obtained in aqueous buffer solutions, the solubility was actually very low and quite constant over the entire pH range, and therefore we wouldn't expect significant drug release in the stomach, which could then supersaturate in the intestine, except maybe under fat state condition the drug release in the presence of lipidic components might already start in the stomach, of course, and then result in potential supersaturation in the intestine.
Thank you. The next question. It is impressive that a linear correlation was observed between predicted and simulated parameters, that is the AUC and the Cmax, for the entire dose range. Despite that nonlinear PK for dose above 120 milligram is seen. Could you please comment on this?
Yeah, I think this is exactly one advantage which is provided by the mechanistic when using the GastroPlus mechanistic absorption model to develop an IVIVC, which as it considers potential limitation due to solubility. So for the higher dosing range, the main factor which becomes limiting for absorption is the limited solubility of the API in the intestinal fluids, and the the IVIVC model actually accounts for this limitation and results in this linear correlation, even if even for the higher dose range, where earlier we observed solubility limitation. Yeah.
Thank you. The next question. Why did you choose to use the sachets instead of a capsule dosage form where the granules would have been loosely packed? Did you have the chance to study the effect of compression force on the tablet disintegration rate that would eventually affect the dissolution rate?
Was supposed to be the most appropriate formulation strategy.
Cordula?
Yeah.
I'm sorry. Seems like you're broken a few seconds. I didn't hear your voice. Did you hear the question?
Well, I heard the question about the granules formulation, why we use the granules formulation instead of a capsule formulation.
Right. Yeah, sorry.
Yeah.
I hear your comment.
Okay, so my answer was that the reason for using a granules formulation in phase 2 was that the purpose was to allow drug administration to patients who have swallowing difficulties, and for this purpose the granules in sachet formulation was the most appropriate formulation.
Okay. Did you check the effect of compression force on the tablet disintegration rate?
Yeah, so the disintegration rate of the tablet formulations was always very fast. So the impact of compression force on the dissolution rate, which we see on this slide, is actually not due to a slower disintegration, but rather to the slower dissolution rate of the API particles.
Thank you.
Mm-hmm.
Did you use in vitro dissolution profile when developing model in granules and the film coated the tablet?
Okay, before phase 2 studies. I assume the question is whether we used in vitro dissolution profiles before going into clinics.
In the time that you developed the okay, the entire question here, did you use in vitro dissolution profile in time of developing model in granules and the film coated tablet?
In the earlier oral absorption model, we just assumed the Johnson dissolution model, which is the default model in GastroPlus, and we didn't directly include the in vitro dissolution data in the oral absorption model. This has been done just later on when we developed the IVIVC model. Then we directly use the in vitro dissolution data.
Thank you. The next question. Is it possible to predict the percolation threshold, or it is only determined experimentally?
Yeah, so of course the likelihood to exceed the percolation threshold increases with higher drug load. However, it is a combination of several factors, so that it is difficult to say at which drug load exactly we are exceeding the percolation threshold. In the case of the present compound, we know that the API itself has very cohesive properties, and this already facilitates, of course, the aggregation of API particles in the formulation and in the tablet matrix. And in addition, due to the compression steps during formulation processing, this problem was further enhanced. So it was really the combination of several factors which resulted then in the exceedance of the percolation threshold. And it's difficult to generalize these to other molecules and other formulations. Yeah, it's specific for this compound.
Thank you. Was the IVIVC only achieved for fat state? Was the GastroPlus simulation showing in this talk all in fat state?
Yeah, right. The IVIVC model development, as well as the data we used for model validation, have been done using fat state data and the corresponding in vitro data in fat state simulated intestinal fluids. And here again, we focused on fat state data only simply because most clinical PK studies were done under fat state conditions, so we had much more clinical data available to verify and improve our models.
Thank you. Next question. How can the information obtained so far facilitate the future formulation development for this drug?
Yeah, so I think, for example, the learning or the fact that we know that there is a dosing range where we observe dissolution rate limited absorption and a dosing range where we observe solubility limited absorption, we tells us that depending on the final dose range of this API, the formulation may have more or less influence on the dissolution rate and therefore on the oral exposure of the compound. Yeah, especially in this case, if future doses will be below, let's say, around 130 milligrams, yeah, much attention has to be paid with regard to API, for example, API particle size or formulation composition and formulation processing, which may then affect the dissolution rate in the GI tract.
Thank you. Can the same model be applied to enabling formulation? That is, for example, amorphous?
Well, I'm not sure if I understood the question correctly. Is the IVIVC model meant here, or?
My understanding is the general gastro model, the ACAD model that established this way. Because
maybe I can chime in the meantime while Cordula is thinking about her response.
Yeah.
One thing that I would suggest that you need to pay attention to is what would be the really the limiting step with the compound and formulations that Cordula was presenting. The critical factors were the rate of dissolution and the solubility limit. Right? With the enabling formulations, the precipitation may start becoming more significant factor if the dissolution rate is fast, but the precipitation, the subsequent precipitation may be the more significant factor in the overall exposure.
Thank you, Vera.
Yeah, thank you.
And it's one related question. Was this necessary to optimize any of the model parameters, or the good fit was achieved only directly using the experimental and the default input values?
Well, in this case, we actually obtained a very good model fit by using the experimental in vitro data. Of course, we had to convert the measured permeability values into effective permeability values to be used then in the model, but there was no need for further parameter optimization to obtain this model fit.
Thank you. The next question, why use a Johnson model instead of Z-factor model? Seems like I lost you again. I cannot hear you, Maduri. No, Cordula.
Yeah, I think it works again.
Can you repeat your answer? Sorry, I didn't hear you.
Okay, could you maybe repeat the question because there was a technical issue?
Okay, why use the Johnson model instead of a Z-factor model?
Well, in that case, we actually directly obtained a good model fit with the Johnson dissolution model, and we didn't try further other potential dissolution models which would be used in GastroPlus.
Yeah, thank you. This is the follow-up from Natalie about, you said you did not submit to the regulatory authorities for your IVIVC. The question, did you plan to do it, or do you plan to do it?
Well, I think we will certainly consider to submit the IVIVC model to the authorities when we will reach this stage. Yes.
Okay, thank you. The next question related to your in vitro, dissol, and in vivo data, 40 milligram versus 120 milligram, slide 21. If the in vitro dissol was done for 120 milligram, so here you're showing 40 milligram, would the method still be discriminating? Was IVIVC still possible?
Yeah, well, if we would use the 120 milligram, the entire dosage form, then we would quite rapidly reach the solubility limit, and then the IVIVC model wouldn't work anymore. So it wouldn't be able to discriminate anymore between the different dissolution rates, simply because the the solubility limit would be reached quite rapidly in vitro.
Okay, the next question. If more excipients are added to the higher dose formulation so that the drug excipient ratio is constant, would you expect a high release from a high dose formulation?
If I understand correctly, the question is whether drug release would become faster if we would increase the amount of excipients, for example, in this case where we have the 120 milligram film-coated tablet. And yeah, I would say that if we increase the amount of excipients, as a consequence, of course, the tablet would become bigger and the drug load would decrease, and most likely the dissolution rate would again become faster due to the release of the primary API particles, as we have observed for the lower drug load tablet formulations.
Now, I guess the question is, if you keep the drug excipient ratio constant at dose, will you expect a high release from a high dose formulation? So you keep the drug loading constant. My understanding that was the question. Because the tablet size limit, you will have to increase the drug loading or change the drug excipient ratio in order to meet the pill size. But now the question is, if you keep the ratio same, that means you give a much larger tablet or you give more number of tablets. Would you still expect a high release from high dose formulation?
Well, I would expect in that case that we may, the drug release rate may be as fast as we observed with this specific drug load, but we may reach earlier the solubility limit, and therefore we will end up, as we have observed for the 250 milligram tablet formulation, in a dosing range where the particle size and therefore the drug release rate doesn't affect drug exposure much significantly anymore.
Yeah, I would agree, because unless your excipients, functional excipients, if you are just the conventional excipients, if you reach a solubility limit absorption, then there's not going to help that much, right?
Right. Yeah, exactly. Yeah. And except if, as you said, if you add some excipients which improve the apparent solubility of the API in the intestinal fluids.
Okay, the next question is percolation threshold related to the solubility of the drug in the excipient solid matrix?
I would say that the percolation threshold is rather related to the API properties like cohesiveness and the properties of the excipients and yeah, maybe the shape of API particles and size and density and so on, rather than the solubility of the API in the tablet excipients composition.
The next question related to this one, what minimum length scale of drug aggregation would be defined as percolation threshold?
Sorry, may I ask you to repeat the question? What minimal?
What minimum length scale of the drug aggregation would be defined as the percolation threshold?
That's a good question. I wouldn't know which length scale would define exactly the exceedance of the percolation threshold. At the end, in our studies as well, we observed the dissolution rate as a consequence of the exceedance of the percolation threshold rather than certain exceedance of a specific size of aggregates in the tablet matrix.
Thank you. There's one more question about the percolation threshold. Were formulations above, formulations above the percolation threshold have a higher variability in vivo in terms of Cmax?
Well, the variability of the in vivo data was actually quite similar to the other formulations. So we didn't observe a significant difference in terms of variability of the oral exposure in these specific formulations, which exceeded the percolation threshold.
Thank you. Next question, is the drug metabolite in the gut?
I have to admit that I'm not aware of any metabolism in the liver, as we mainly focus on the pre-absorptive part of the pharmacokinetics of this compound. Yeah, and yeah, whatever was related to clearance and disposition was was described using the intravenous data.
Okay, we've got the last question so far. Is the IVIVC model overruling the Johnson dissolution model? I assume that the dissol rate is based on the dissolution profile instead of particle size when you use IVIVC. Is that correct?
Yes, right. So,
oh, I can now hear you now again.
Cordula, we've lost the audio again.
I can hear you now.
Okay.
Can you, did you hear my question?
Yes, I heard your question. So,
could you repeat your answer, because we didn't hear you?
Yeah, so actually in the IVIVC model we use the in vitro dissolution profile, which accounts for the properties of the entire formulation, and while in case of the Johnson dissolution model we consider the specific properties of the API, and yeah, this I guess is at the end, the difference we observe between the result we obtained in the IVIVC model and the comparatively worse prediction when using simply the Johnson dissolution model for the film-coated tablet. Because, yeah, we do not consider additionally formulation-related impact on the dissolution rate.
If I could just follow up on this answer, just a quick additional comment. The difference is, right, the Johnson model worked nicely for the granules and for the tablet, where the dissolution was mainly driven by the API particle size. And as you have shown for the film-coated tablet, that has broken, so it wasn't just the API particle size that was determining the rate of dissolution, but it was the formulation itself, and that necessitated to create the IVIVC to find that additional relationship due to the formulation aspects rather than just the particle size alone.
Thank you. Okay, we have completed all the questions that came up so far. Let me look at if anyone raised their hand here, just make sure we address all the questions. Nope, I think we have done all the questions, and also saw that we getting close to the end of the time for our webinar. Well, thank you again, Cordula, for answering these questions, and also Vera for help out answering these questions. In case for the audience, if you have additional questions after the webinar, then please, you can go to our GastroPlus user group LinkedIn website, where you can post your questions, and they will be addressed by our members or members in simulations class. So thank you again, Cordula, for the presentation and answering the questions. For the audience, thank you all for your participation. You may now disconnect from the webinar. Thank you, everyone.